So nucleotides are monomers for DNA, and so nucleotides are made up of 3 different parts. They're first made up of a nitrogenous base, which is either guanine, adenine, thymine, or cytosine. And then nucleotides are also made up of a 5-carbon sugar, which is attached to a phosphate group. So here are the 4 different kinds of nucleotides. There are adenine and guanine are both purines, and that means that they have 2 rings around their nitrogenous base. And cytosine and thymine are pyrimidines, which means that they have 1 ring around their nitrogenous base. So the base pairing rule is that is between adenine and thymine because they can form 2 hydrogen bonds, which you can see in the little niches, and then between guanine and cytosine, which form 3 hydrogen bonds, which again you can see on the
So this is the overall structure of DNA. It forms a double helix, and there's 2 strands which run anti-parallel to each other. And each strand has a 5' end and a 3' end. So the 5' end always has a phosphate on top, and the 3' end always has an OH on top. So those are the 2 strands, and then the backbone of the double helix is made up of phosphates and 5-carbon sugars, and the 5-carbon sugar is called deoxyribose because it lacks an oxygen, and the rungs of the double helix are formed by pairings between the nitrogenous bases. So guanine and cytosine are always paired together, and adenine and thymine are always paired together, and they are bonded by hydrogen bonds in between.
Alright, so DNA replication is a semi-conservative model, meaning that half of the DNA is conserved when you create new DNA strands from the original strand. So the DNA is split first with helicase, which goes through the original strand and breaks it up, and then there is a single-strand binding protein which stabilizes the new strand. On the leading 3' to 5' end, you see that there's DNA polymerase 3, which continuously attaches new DNA to the leading strand. But moving on to the lagging strand, it is synthesized discontinuously because it goes in the opposite direction, 5' to 3'. And this creates, so primase first synthesizes the short RNA primer, which is extended by DNA polymerase, and then creates like an Okazawa fragment, because again, it's not continuously synthesized. And then another DNA polymerase comes through, and that's what replaces that RNA primer with the actual DNA. And then this DNA ligase then goes through and like a glue stick and joins together that replace primer with the Okazawa fragments to create one continuous strand of DNA on the lagging strand.
So helicase then forms a replication fork that splits the 2 parent DNA strands.
On the lagging strand, DNA polymerase 2 acts as a proofreader to ensure that the DNA is replaced accurately and without mistakes.
Protein synthesis starts with the synthesis of RNA, and the transcription of RNA is directed by DNA. So as you can see here, the promoter is what initiates this transcription, and the DNA strands are split apart by the RNA polymerase, and the pre-mRNA come in and pair up with the bases of the DNA.
The backbone of RNA, unlike DNA, is not composed of deoxyribose, but rather the sugar ribose.
RNA transcription continues until the terminator signals the end of transcription, and then the pre-mRNA goes on to RNA splicing.
After the RNA synthesis on a DNA template and before the mRNA leaves the nucleus, first a 5' is formed to protect the RNA from degradation of hydrolytic enzymes, and during protein synthesis they are also used to tell ribosomes where to go. closer to the microphone.
So after that, there's a poly A tail that attaches onto the 3' end, and the poly A tail facilitates RNA export from the nucleus. It inhibits the degradation of RNA in the cytoplasm, and it helps RNA attach to ribosomes. And after that, the RNA moves from the nucleus to the cytoplasm.
In protein synthesis, the mRNA attaches to a ribosome, and the tRNA that is produced picks up specific amino acids through the process of amino acyl tRNA synthetase, and then they line up by means of their anticodon triplets at their mRNA codons, and a codon is basically a nucleotide triplet that forms amino acids.
After the tRNA anticodons line up with mRNA codons, the amino acids attached to tRNA can form a polypeptide. And the 3 sites of the ribosome are the A site, which is where the mRNA enters, the T site where transcription occurs, and the E site where the mRNA exits.